I think. So
yes,
you
it's
can
working
see the screen, and if not, someone will shout. Right?
Yes, it's perfect. Thanks.
Perfect. Thank you very much. So yeah, thanks for the invitation. First of all, I'm happy to be talking here today about genome editing for precision breeding in aquaculture. My name is Diego. I'm a group leader at the Roslin Institute. I'm actually involved both in AQUAIMPACT and AQUA-FAANG, but today I'm going to talk from the AQUA-FAANG side. So I'm a work package leader, co-work package leader, together with my colleague Luca Bargeloni in AQUA-FAANG. He already talked about what we did, I think, in the previous webinar. So today I'm going to talk more about what's the future, right, and how we exploit the results that we have been doing in AQUA-FAANG, but also AQUAIMPACT. So Antti just gave a really good overview about genomic selection and selective breeding in aquaculture and future avenues to improve in this area. Selective breeding basically is now routinely used to improve aquaculture stocks in many aquaculture species around the world. And it basically consists in let me put the pointer. It basically consists in generation after generation, making relatively small, medium improvements in a trait of interest on various traits of interest at the same time, so that the cumulative improvement after a few generations is really quite noticeable. So genome editing is not an alternative, but a complementary approach where the benefit is that you can get really large gains in a single generation. So, compared to selective breeding, which takes quite a few number of generations to build up, that can, in just single generation, get big gains in whatever traits you're interested in. As I said, it's not either or. They are perfectly complementary. They complement each other. Just very briefly, for those, I have a figure here about the CRISPR-Cas system. I assume you are all now very familiar with what CRISPR-Cas9 is and genome editing. CRISPR-Cas9 is the last system that was invented or popularized to genome editing. It comes from bacteria, and I'm sure you've heard about the Nobel Prize recently. It's basically a tool that allows you to edit the DNA of a species, either by creating problems, sort of breaking the DNA and creating errors that for example, may mess up with a protein and disrupt its function. Or by that, on the other side, you can also do direct changes. So you can create a break in the DNA and then try to give it a template so that you basically guide the repair system of a cell to introduce the changes you want in the DNA. Right. So that's genome editing, which I'm sure all of you are familiar with. And this is basically a way of changing the DNA of a species or individual population, right? And as I said, it's the perfect complement to selective breeding. It allows to have large genetic gains in a single generation. That's very relevant for traits that are not easily measured. For example, things like feed efficiency in aquaculture, they are really tricky to measure. And so therefore, it's difficult to implement them in breeding programs. They are independent of existing genetic variation, while selective breeding depends on basically how much genetic variation for the trait of interest exists in the population. Genome editing doesn't depend on that. It can improve traits with very low or no heritability. And it can also help fixate traits. And for example, think about disease resistance. You could create, if you're thinking about the viral disease, if you remove the receptor for a virus, you can basically make the animal directly fully resistant to that virus, right? And that's really useful also for breeding programs, because breeding programs, the more traits you try to focus on in your breeding program using selective breeding, the less progress you make in each of them. So being able to remove traits from that breeding program using genome editing and allowing the breeding program to focus on the other traits can have a massive effect on the rate of genetic gain. Obviously, not everything is pretty and perfect, and this is what I'm going to talk about most of my talk, is the constraints to the implementation of genome editing. First of all, it requires knowledge of the genome. We need to know what we want to edit, where AQUA-FAANG plays a big role, and I'm going to talk the most of my time. It also has technical challenge in how we incorporate genome editing into aquaculture breeding programs. How do we actually make it compatible with running a breeding program and with protecting the environment? And then ethical considerations also, obviously, which also includes the regulation, right? So first, how do we find, on the first topic, how do we find what do we want to edit? We can edit things, but we need to know which regions in the genome, which pieces of the DNA, which has 3 billion, between 500 million and 3 billion pieces, which ones do we edit? Which ones do we change? How do we change the genome so that it produces the change that we want in the phenotype of the animals, make them grow faster, make them more resistant to diseases. And this is generally not really focused on, at the moment, in specific base pairs in the DNA, but rather in genes, but that's still 20,000, 25,000 genes in each species. So that's huge, right? A huge number of genes to investigate for every single trait. And that's where AQUA-FAANG comes in, as I was saying, where we have been building really maps of the genome for the 6 most important European aquaculture species. Not only reference genomes, high quality reference genome, but also where the genes fall, what are the regulatory regions, how they respond all this in the face of, for example, the viral challenge or the bacterial challenge. And this is a fundamental piece that will underline lots of research in the future years to try to basically find these pieces of DNA. In our research group, we follow generally three avenues to try to find these pieces of DNA, genes that we want to edit. One of them is around understanding QTLs, existing population. Another one is about using comparative genomics. And finally, the last one is about using pool CRISPR screens. So I will go relatively quickly through each of them, starting by the one that you may be most familiar with, and much intuitive. So it's about finding QTL from the regions in the genome that are involved in difference for a trait and trying to fixate that variation, right? As Santi was alluding to in his talk. So here I have an example that actually Luca Bargeloni presented in the last webinar. It's about resistance to VNM in European sea bass. We did basically a genome-wide association analysis where we found a major QTL in linkage group 12 of this species, which explains high percentage of genetic variation. And we found actually 2 genes that overlap with that QTL, pointing directly to these genes as clear candidates to improve resistance to that disease. The actually animals that have the resistant version of whatever is causing resistance, which we will here resistance allele, are practically fully resistant to the disease. So via genome editing, via incorporating that variation into the genome of a population, we could basically create fully resistant European sea bass to VNM. That's a way to get those edits that you want using GWAS and QTLs. Another example is using comparative genomics, basically getting from a species or even just populations, different populations of the same species that have a trait that is desirable, trying to move that across to a different population or a species. So, for example, a really iconic example is that of salmonids and sea lice. Sea lice is the biggest problem currently in Atlantic salmon aquaculture. Atlantic salmon is highly susceptible to sea lice, these copepod parasites that attach to the skin and feed off the blood and mucus. But coho salmon, a sister species or another salmonid species, is fully resistant to sea lice. The lice still attached to the coho, but they are able to mount an immune response that basically rejects the lice. So what we are doing is using basically single cell genomics to try to understand that complex response that coho salmon has. Here I'm showing a single cell RNA sequencing of the coho immune response to the sea lice, where we have annotated different cell types. And yeah, we are trying to basically guess what's the mechanism underlying that resistance, with the view of within this project, which is actually a collaboration with many partners, including a big one leading some of the efforts of the projects in this area, Nick Robinson from Nofima. Yeah, this is what we are trying to do, basically. Then find out what causes resistance, transform it, and put it into Atlantic salmon. So again, comparative genomics, another way of getting those targets that you need and you want to use. And there is a final one, which is really interesting, has huge potential, which is basically why, depend on what you observe in population or species, you can just generate new desirable variation with genome editing. That what we are doing here at the Institute using cell lines for various viruses, we are generating what is called a CRISPR screen, a pool CRISPR screen, where you basically use guides that are targeting every gene in the genome of a species. And in a cell culture, you knock out all those genes at the same time, simultaneously, but one knockout in each cell. So there will be a pool of cells, millions of cells, with each of them knockout for one of the genes in the genome. And then you can apply a pressure, which in our case, a viral challenge, and see which ones survive, see which knockouts, which cells that are lacking one gene are able to survive the infection. So for example, if one of the genes you are knocking is probably the receptor that the virus needs to get into the cell. If you do that, the virus won't be able to get into the cell, making it fully resistant. You will be able to pick it up with this unbiased screen and genome-wide screen. The second alternative, second version of this, creating new variants, rather than making it randomly or unbiased, you can trust what you know about biology. This is an example from Roslin in pigs. Pigs and the virus PRRSV, porcine respiratory and reproductive syndrome virus. In any case, anyway, virus. So the researchers here knew that the virus binded the exon 7 of this gene, CD163. So what they did was knocking out that exon, something that doesn't exist in generating something new that doesn't exist in nature, to basically make the pigs fully resistant to disease and solving the problem. So this is the three. I'm sure other people may come with other ways of classifying this or other new methods. These are the ones that we are using at Roslin to get our targets, and then with the idea of doing genome editing in our fish, and then hopefully eventually implementing it in commercial aquaculture. And that's the next problem and implementation. We focus more as scientists and researchers in the first phase, the discovery, the understanding the genes, what do we have to edit, but the implementation is as tricky. Where do we edit our animals? Do we edit our stocks, making them genome editing forever, so to speak, or do we actually keep our stock unedited and do the Edison multiplier or production phase? If we decide to go for that, how do we deliver genome editing en masse at high efficiency? At the moment, most of our editing requires microinjection of eggs, which is very laborious and very manual. So do we need to generate new systems to do that much more massively and efficiently? Mosaic systems is another problem, that if you are trying to do the edits in the fish that you are going to produce, at the moment we have lots of problems, not problems, but we see that a fish may carry different edits, and the edits in some of the cells, they might be edited, and some of them may not. Again, we need optimization. We need to control for off targets, although this is relatively straightforward, but just sequencing the genome of the animals that we are editing. And finally, how do we control the interactions with wild populations? Livestock don't have this issue, but in aquaculture, this is very much a very important issue. We have wild populations, and if we are generating genome-edited fish and putting them in pens in the oceans, they will interact with wild populations. How do we control for that? And that's the third challenge, I guess, of genome editing, right? That's the ethical considerations. When should we apply genome editing? I should have put here and how, right? And there are many things to be considered, I believe. We are working with Nick Robinson, again from NOFIMA, in a way of trying to consistently and objectively analyze whether we should do an edit and implement it as based on basically what's the benefits, not only to the animals in terms of welfare, but also to the society, but also what are the impacts, right? How does it impact the greenhouse gas emissions? And very importantly, how does it connect with the local ecosystems? As I said, if we are putting genome-edited animals in the wild, it's very likely that the genome-edited animals will have to be sterile, right? And if so, going back to the previous point, is that sterile animal that we are producing compatible with the breeding program, right, and continuing to select our animals every generation. So there is a lot of technological innovation required, a lot of, but also quite a few ethical considerations to be made by producers and breeders, right, about when an edit is necessary and desirable. Nonetheless, there has been lots of applications of genome editing of cultured species, many in cell culture, but also a few, or several in vivo, by many different species, as you can see, and for different traits. Many focus in growth, which is probably the easiest one, but also many in sex reproduction. Things like disease resistance and immunity are lagging behind, although we have had some success in this area. They are coming also into production. So Japan has, this was news from 2021, where Japan already has approved for commercialization to genome-edited fish. One of them is edited for a leptin receptor, making them, it's fugu, making them just grow faster because they eat more. And another one, is knocking out the myostatin gene in red cyprim, which basically increases the feed efficiency. And here it ties with what I was saying about the connecting with breeding programs for traits that are very difficult to select, but also with thinking about when we do the edits, improving feed efficiency is really beneficial for everything, for the environment, because the feed, well, they require less feed. We are reducing greenhouse emissions. We are producing more with less. Whether making the fish eat more by knocking out the leptin receptor is as desirable, is something to consider when thinking about what we want to edit, right? And I want to end just with saying, before the conclusions, that the potential of genome editing is very clear, right? We can improve how we feed the ball, producing more with less. We can improve animal welfare, reducing the transmission of diseases, increasing the resistance of diseases in our animals, making them more healthy, and also avoiding the transmission of diseases to the environment, which involves reducing environmental impact, both of them feeding the world more efficiently, making our animals more efficiently and more resistant, both contribute to reducing the environmental impact. And finally, they can help improve human health, not only by, for example, in changing the nutritional components of our animals, but also by reducing the need to use antibiotics and reducing, therefore, antimicrobial resistance in our food system. So I will finish with quick conclusions. As I was saying, genome editing is a great complement to what we are doing in selective breeding and to AQUAIMPACT has been focused on. We can generate large gains in single generations. It's independent of the variation that exists in the populations. And there it allows removing traits from selection indexes in breeding programs, allowing them to focus on other things. It is heavily dependent on the targets. We need to find targets, what we need to edit, discover, introgress, or create new disease-resistant alleles, which have, well, disease-resistant or other traits that can lead to big wins. And the functional genomics and new technologies are like the ones using AQUA-FAANG are integral to this discovery of suitable targets. And finally, there is a lot to be considered still in how we implement these edits and also whether we implement them or not, right? And that requires a lot of research, but also thinking and dialogue. Genome editing is already happening, it's coming, it's there. And possibly I would say that regulation and dialogue with policymakers and society is lagging behind and something we need to work on. Nothing else, acknowledge the loss of people involved in the work that I presented. Obviously, AQUA-FAANG, and first of all and foremost, my team. All what I present, and it's all thanks to them. It's their merit, and yeah, they are great. So thank you to them very much.